Cell cycle control system. Cell cycle control system. The cell cycle control system checks cells to ensure that important processes, such as DNA replication and repair, are working properly before division. It ensures that damaged cells are not allowed to divide. Three major checkpoints exist to regulate the cycle at the end of gap 1, the restriction point, at the gap 2, M transition, and during M, spindle assembly checkpoint. At each checkpoint, if cellular surveillance systems detect a chromosomal defect or a metabolic problem, the checkpoint is activated. Once activated, the cell will stop in the cell cycle and repair processes begin. If repaired, the cell is released from the checkpoint and continues through the cell cycle. If the cell cannot be repaired, a death program is initiated and the cell dies rapidly. G0 phase. Fully mature, functioning cells leave the cell cycle and carry out their specialized functions in G0 phase. If they re-enter the cell cycle, they will pass through the G1 checkpoint and into S. Examples of cells that are almost always in G0 in adults are neurons and muscle cells. Cells that are not ready to divide or have a defect should get stopped and repaired at G1, G2, or during M. But what if a defective cell is not stopped, is not repaired, and does not die? Cancer is a general term for many diseases in multicellular organisms that are caused by uncontrolled cell division. Cancer cells and normal cells are identical, with the exception that cancer cells divide uncontrollably. Cancer cells do not get a positive signal to proceed by the cell cycle control system, but continue to reproduce anyway. Cancer cells can metastasize or spread to other sites in the body. Contact inhibition. Cells typically will only grow and reproduce until they touch each other, and then the cell cycle control system will stop signaling the cell to proceed. This is called contact inhibition. Cancer cells do not exhibit contact inhibition. Instead, they grow into masses called tumors. Some cancer cells continually synthesize factors that keep them dividing. Click on the link to view a video on contact inhibition. Cancerous tumors. Tumors that cause damage to surrounding tissues are called malignant tumors. They are also said to metastasize, systematically spread the cancer to other areas of the body, usually by way of the bloodstream. Tumors that are not life-threatening or otherwise damaging are called benign tumors. In this picture, we have oral carcinoma cells. This mass of cancer cells right here do not have any kind of contact inhibition, so they just continuously grow into a crowded ball of cells. Cancer. Typically, when someone dies from cancer, it is not the result of the primary tumor, but instead the metastases, or the secondary malignant tumors, are the cause of death. General types of cancers. Carcinomas are epithelial tissue cancers. Epithelial tissue, that's an anatomy term. Epithelial means surface coverings, which includes skin or coverings of organs. And sarcomas are connective tissue cancers. Connective tissue is what the word implies. It's tissue that connects organs together and sort of provides support for other tissues. And leukemias, lymphomas, multiple myeloma, these are cancers of blood-forming tissues. One specific type of cancer, melanoma, is a carcinoma, or a skin cancer. This type of cancer is a very concerning cancer because it's very dangerous if it metastasizes. The good news is, if caught early and as it metastasizes, the outlook is very good for a cancer patient. What dermatologists use to evaluate moles, or these specific type of skin lesions, is the ABCDE method. They look at a set of conditions to determine if a mole is possibly a melanoma. A stands for asymmetrical skin lesion. So as we can see in part A of this diagram, the moles themselves aren't too symmetrical. And B, the border of the lesion is irregular, so the edges aren't smooth, they are a little rough. C, color. Melanomas usually have multiple colors and not just brown. As we can see in this diagram here, part C, there's a little bit of green in there as well. D. Diameter. If the mole is greater than the head of a pencil eraser, it could possibly be a melanoma, a couple millimeters in diameter, and E. Enlarging or evolving, changing over time. If any one of these conditions is present, it is suspect that the mole may be a melanoma, but of course it's always best to see your dermatologist to evaluate the mole, as they are medically professional and qualified to determine these things. Treatment of cancers. Chemotherapy and radiation are the two most prescribed treatments for cancers. In the diagram on the left, we see a bottle of a chemical, a drug, that will be infused into a patient as chemotherapy treatment. And in the diagram on the right, we see a patient undergoing radiation treatment. Chemotherapy and radiation. Chemotherapy disrupts the cell cycle, typically targeting the mitotic spindle formation. Chemotherapy is systemic, that is, it affects the whole body. Radiation is location specific, directed at the area affected by the tumor. It disrupts the cell cycle by damaging the DNA in the area, and the cancer cells cannot repair themselves and will stop dividing. Side effects of cancer treatment. The typical side effects from chemotherapy are from the damage occurring to the normal cells, which are affected by the chemicals. This is seen easily in the fast reproducing cells, like the hair follicles, causing hair to fall out, and of the digestive tract, causing nausea. So, since cancer is a fast reproducing cell, these drugs are designed to attack those cells, but unfortunately it comes at a cost, also affecting the normal cells that are also fast reproducing. Bone marrow transplants. Blood cancers are also treated with bone marrow transplants. This involves a surgical procedure where bone marrow is removed, usually from the pelvic bone, and transplanted into the cancer patient. A patient may serve as his or her own donor in some cases. This treatment is used for blood cancers because bone marrow produces stem cells, unspecialized cells, that can divide through mitosis and differentiate into diverse specialized cell types. These cells can produce new, non-cancerous blood cells. Stem cells. In mammals, there are two types of stem cells. The first type is embryonic. These are found in the blastocysts of developing embryos, and embryonic stem cells can differentiate into any type of cell, any organ, any tissue. Potency is pluripotent. This means that they could differentiate into any cell type in the organism. Then the other type of stem cells that we have are adult stem cells. Adult stem cells act as a repair system for maintaining the turnover of regenerative organs such as blood, skin, and intestinal tissue. So unlike embryonic stem cells, these adult stem cells cannot make any tissue or organ in the body. They can only do a few different types, but are mainly for replacing the tissue that is lost for regenerative tissue. The potency of these cells is multipotent. They can differentiate into some, but not all, cell types present in the adult organism. So for example, an adult stem cell could be one that could differentiate into muscle, tendon, or bone. Stem cell technology. In addition to cancer treatment, scientists are developing methods for using stem cells to treat other ailments. Remember, since stem cells can produce many different kinds of tissues and organs, it can be used in replacement treatments for organs that are damaged and need to be replaced. A trachea or windpipe pictured here on the left was grown from harvested adult stem cells, and it was used to replace a woman's damaged windpipe. Because the stem cells were her own, there was no chance for rejection by her immune system. Embryonic stem cell technology. Presently, embryonic stem cells have been used primarily for research. Potential for technologies exist, but currently no product has been produced. Promising research using embryonic stem cells can include tissue engineering for organ transplants, in vitro models to test drug response and predict toxicity, creation of neurons for the treatment of Parkinson's disease, and alternative treatment for diabetes.